Thanks a lot for attending today. This is the PDHP workshop, Tools for Reproducible Research with Alexander Cernot. My name is Paul Schultz. I'm a data scientist and statistician here for the Population Dynamics and Health program here at the University of Michigan in Ann Arbor, Michigan. I just want to take a couple minutes before we get started and just kind of plug our center real quick and kind of talk to you about the resources that we've got on offer, and then I'll hand over to our speaker in about 3 or 4 minutes here. So you can see I've got our website up here. It's pdhp.isr.umich.edu. Obviously, if you RSVP'd for the workshop, you've at least touched the workshop page. So this website is kind of the home for our center. We're a center supporting population research, backstopped by the National Institute for Child Health and Human Development. And we're charged with sort of supporting all aspects of population research, especially with a particular focus on early career scientists. And so we've got a multiple course center here. We do all sorts of resources for our affiliates and also some free resources for those that aren't affiliated. We've got a career development program where we have a formal mentorship program where you're linked up with a successfully funded NIH scientist and to develop your career. And then we also offer a small grants program here. You can see a tab for that. So we disperse funds throughout the year to support research. Anything, any research needs could be supported through that small grant program. So computing hardware, for example, but also just funds to collect population research of any kind. So I encourage you to take a look at that. We also do paid client project work as well. And so we've got a team of technical folks here, myself, Liz Hanley, and Hu-Chen Liu, under the direction of Dr. Brady West. We offer free consulting to our affiliates, but then we also can deploy our analysts out to your project as well on a paid basis. So if you do have research needs that you're unable to hire on your own, or you just would rather sort of, hire on a fraction of an analyst, that is a service we offer as well. And again, you can reach out to me for that. So that's just a real quick thumbnail overview. Again, the website is pdhp.isr.umich.edu, and I encourage you to take a look at that. If everything's outlined there. If you've got questions, please feel free to reach out to me via email or on the side as well. One last thing I'll say is that we've also got a weekly coder space. So our technical team hosts sort of an office hours online via Zoom. It's Tuesdays 2 p.m. to 3:30 p.m. Eastern time. And again, you can find the link to that. Our team hosts a Tuesday session, but there's sessions multiple times throughout the week. So that's available. The information for that is at datascience.isr.umich.edu. So that's a quick plug for our center. Again, if you've got questions, please feel free to reach out to me. Just wanted to point out that, if you're only familiar with our workshop series, we have other resources on offer that you can take advantage of. So focusing on today's workshop, this is part of an ongoing workshop series. I'm not unfortunately able to announce another upcoming workshop right at the moment, but this is an ongoing series that we'll have throughout the year. So this is our second one of 2022. And again, thanks a lot for coming today. Keep in mind that the workshop is recorded. So by default, please make sure that your video and audio are muted. The chat session is open, so I encourage you to chat in your questions as opposed to video and audio. And of course, if anything sort of weird or embarrassing happens on your end, it will be recorded. So just be aware. Also, the reason that we do record is so that we can post the video online later. And this is a common question I get all the time. And so yes, the video will be posted to the website after the workshop. We've had trouble with some of the past workshops. So you'll see that not all the videos for the past workshops are online, but we've closed that loophole off. And so going forward, you should have video for all these workshops online, really for all time going forward. So that's my preamble for the workshop. So again, thanks again for coming. I'll introduce our speaker now, and then we'll be off and running. So our speaker today is Alexander Cernot. He is an associate professor in the social statistics department at the University of Manchester. He has a PhD in survey methodology from the University of Essex and was a postdoc at the National Center for Research Methods and the Kathy Marsh Institute. His research and teaching focus on survey methodology, longitudinal data, measurement error, latent variable modeling, and new forms of data and missing data. And you can find more about him and his research at www.alexcernot.com. So again, thanks to our speaker, Alex, and I'll hand off to you. Thank you.
Hello, thank you so much for having me. So today I want to talk a little bit about reproducible research. And this is a really important topic. In the social sciences, I think it kind of got a lot of attention in psychology, but at the same time, there's lots of work happening in kind of in health research. And more people in the social sciences are aware that this is a problem. So I want to kind of present the overall problem and some possible solutions. And then towards the end, I'm going to talk about more technical solutions to this problem, talking about dynamic documents and version controls and things like that. So we'll cover quite a lot. You can think of this as a introduction, and at the end I also have some references if you want to go and learn more. So before we start, it might be useful to think again what we're doing here. So what's the aim of research and science in general, and then we can discuss about what are the issues and possible solutions to that. And for this, I think it's good to think of 4 core values that we should have as scientists. And these were the ones proposed by Merton. And I'm going to go through each one and just give you an overview of what it means. I think most of you are aware of these and probably agree with them, but it's good to be reminded of them from time to time. And then we'll discuss how maybe what's happening now does not really fit with these core values. So the first core value is universalism. And the idea here is that really anyone should be able to do science, do empirical work, to propose hypotheses and investigate them. And it shouldn't really depend on the status or where you come from, or if you're a PhD student or the president of the International Statistical Association. In principle, all of the people that are part of the kind of science framework and are doing research should be able to have the same say to propose hypotheses and to test them empirically. And it shouldn't depend on any of our statuses, how much we trust the results, and how, if something is truthful or not, should not depend our status in our field. The second one is communality, and the idea here is that a lot of the research we're doing is really built on what other people have done, or research that is done collaboratively with other people. So we have to be aware that actually we're not working on our own, even if we think we invented something or we came up with something very innovative. It's probably based on the work that other people have done. And another important thing that is related to this is this idea that in order to do this work, we actually need to be able to see what other people have done and be able to be part of an open community where we see the research done by other people and are able to investigate maybe the code or the data that other people have done in order to move science forward. So the idea of doing things together and being part of a community also implies that we should be more open in sharing our code and our practices and our data, and to accept the fact that we're part of this group that needs to work together in order to move science forward. The third principle is the lack of conflict of interest. So the idea is that as scientists, our main aim should be to uncover the truth. So it shouldn't be, to sell products or to only look at evidence that supports our pet idea. We should be open-minded, and we should really think of what is the truth and less about other possible interests like our career or our company or our institutions. While those are important for us, for science, they are less important. So our focus should always be for to kind of find the truth, regardless of the implications for our own career or institutions. So again, I think most of us would agree with this, although again, it's not always obvious that this is happening in the real world, because people feel different types of pressures when they do research in academia and also outside. And finally, this idea of organized skepticism. So the idea is that we should be able to investigate any kind of question. There shouldn't be limits about what we can investigate and we cannot investigate, and we should critically assess the research that other people have done. So again, if it doesn't really matter who the theories from before, who they come from, it matters that they're open theories that could be empirically tested, and again, we should be able to critically assess all the statements that are made regarding science. Okay, so all of these I think are pretty general concepts or values that I think most of us can agree with. But the issue is that even if in principle they sound fine and we don't have any issues with them, often the reality is more messy, and because of different reasons we might not be able to meet up or to respect these values. What I'm going to do is I'm going to show you two short videos. One is about how scientists perceive science and issues that are currently are problematic with scientific research. And then I'm also going to show you a short video about what this could mean also for what the general public thinks about science. Okay, so hopefully you should be able to hear and see this.
Is there a reproducibility crisis in science? Nature asked 1,500 researchers this question as part of an online survey. Half agreed there's a significant crisis, and many more identified a slight crisis. Science moves forward through corroboration, when researchers verify others' results. But 70% of researchers who took our survey said they'd tried and failed to reproduce another group's experiments. And even when trying to reproduce their own work, just over half said they couldn't. Does this mean that most published research is wrong? Our respondents don't think so. Most still trust published results in their field, with physicists and chemists the most confident. There are many reasons why researchers may fail to reproduce results. Sometimes a small difference like shaking or stirring a sample can lead to different outcomes. But the biggest reason, according to our survey, is selective reporting. Things like cherry-picking data to support a pet hypothesis. To get to grips with reproducibility, we need better mentorship, proper use of statistics, and more robust experimental design. All these approaches got ringing endorsements in our survey. Whether it's a crisis or not, right now lots of people are paying attention to reproducibility. That makes it a good time for everyone, funders, publishers, scientists, and the institutions they work for, to act. Read more and delve into the survey data at nature.com/news.
Okay, so that was the first video. So this kind of concentrated on how researchers or scientists see the practice of science currently. The next, there's a video about how the public might see science and the issues that are currently present.
Science, the thing we love and respect so much, we only allow scientists to be portrayed by the likes of Arnold Schwarzenegger, Nicolas Cage, and Al Pacino. That is how much we respect them and the complexity of the work they do. Science is constantly producing new studies, as you would know if you've ever watched TV.
A new study shows how sugar might fuel the growth of cancer.
A new study shows late-night snacking could damage the part of your brain that creates and stores memories.
A new study finds pizza is the most addictive food in America.
A new study suggests hugging your dog is bad for your dog.
A new study showing that drinking a glass of red wine is just as good as spending an hour at the gym.
What? That last one? No! That last one doesn't even sound like science. It's more like something your sassy aunt would wear on a T-shirt. Time.com once even ran the headline, "Scientists say smelling farts might prevent cancer." It turns out the study never mentioned either of those things. It just pointed out that certain sulfide compounds are useful pharmacological tools to study mitochondrial dysfunction. And while that Time story was later heavily corrected, the scientists told us that we still get phone calls and emails from strange radio and TV shows wanting us to talk about farts. which is clearly a waste of their time. There are now so many studies being thrown around, they can seem to contradict one another. In just the last few months, we've seen studies about coffee that claim it may reverse the effects of liver damage, help prevent colon cancer, decrease the risk of endometrial cancer, and increase the risk of miscarriage. Scientists are under constant pressure to publish, with tenure and funding on the line. And to get published, it helps to have results that seem new and striking, because scientists know nobody is publishing a study that says nothing up with acai berries. And to get those results, there are all sorts of ways that, consciously or not, you can tweak your study. You could alter how long it lasts, or make your random sample too small to be reliable, or engage in something that scientists call p-hacking. It's very complicated. But it basically means collecting lots of variables and then playing with your data until you find something that counts as statistically significant but is probably meaningless. For example, the website 538 surveyed 54 people and collected over 1,000 variables, and through p-hacking the results, was able to find statistically significant correlations between eating cabbage and having an innie belly button, drinking iced tea and believing Crash didn't deserve to win Best Picture, and eating raw tomatoes and Judaism. And if you think I'm exaggerating about the impact that this misreporting can have on our faith in science, look at an example from some of the people most guilty of it. Because the Today Show, which lives for scientific studies, recently concluded one segment like this.
Like a lot of studies that we love around here, there have been a couple, especially related to women, about the benefits, as I get all serious, on whole milk. Hey, whole milk, but it's true.
Well, there's a lot of research, though, that says actually having whole milk or having whole fat dairy products actually can help you lose weight.
I think the way to live your life is you find the study that sounds best to you, and you go with that.
No! No, 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 no,
no! Okay, so hopefully that drives home the point about, you know, the fact that what we're doing as scientists is very important for how the public sees science and, you know, for the future of our endeavor. So in the next few hours, I'm going to talk about the problems that were already a little kind of mentioned in the previous videos. Then about some of the solutions, kind of the high level solutions that are being considered. And then I'm going to discuss a little bit about the practices, or, you know, kind of smaller things that you can do to improve your work with data and increase the reproducibility of your work. Okay, so let's start with the problems. So one of the main problems is this idea of kind of p-hacking, or kind of selective reporting. And this, I found this comic quite illustrative of this. So imagine we have a research question such as what's the relationship between jelly beans and acne, if jelly beans are causing acne or not. We want to investigate this. And let's say that we do some research and we find no relationship. But then we have another hunch that maybe, well, maybe it's a particular color that is the cause. So we should look at the effect of jelly beans on acne for different colors. And we go about doing this many times, and then eventually one of these tests is significant. So here we see that out of 20 tests, we find one that is significant. And we see that there's a relationship between eating jelly beans and acne if you eat green jelly beans. Okay, and then the problem is that only part of these results end up being published, and then we might have this kind of news report about how green jelly beans is causing acne and how very confident we are about these results. And this kind of high-level issue of p-hacking, we can think of it as two parts. Or on the one part, we have an issue with publication. So the issue is that basically only certain articles have higher chances of being published compared to others. And this is also known as publication bias. And then we have this other issue of specification search. So the fact that researchers also tend to have a bias in selecting only certain results to share with the public and to publish. So let's start with the first one, with publication bias. So there's quite a lot of research and evidence at this point that there are certain studies that are more likely to be published. So if the studies are using new kind of data, if they find unexpected results, if they find significant results, then they are more likely to be published than if you're doing replication studies, if you have a null result, and if you're not doing something innovative. So because of this publication bias, then we have an issue with what is actually reported and what we think as the truth in the real world. So another issue related to this is also the fact that researchers kind of on their own select what they want to publish. So on the one hand, the review process is blocking certain types of articles, and on the other hand, researchers on their own are selecting only some articles to be written up and submitted. This is also known as the file drawer problem. So if you do some research and you're not convinced about the findings, or you find something unexpected, you might be less likely to publish and share your results with others. And again, this is very useful information that is lost just because of this self-censoring of researchers. So a nice way to find out about this process is to look at places where you have to pre-register your research. So a good example of this is the time-sharing experiments in the social sciences. So this is a study where researchers can submit an idea, and then if they get in, data will be collected to investigate that research idea, and then they can use the data and publish the results. And so here we know the baseline, we know all of the studies that were accepted and for which data was collected. And also we can investigate to see how many of these were actually published afterwards. And what we see is a very clear pattern, a relationship between how strong are the results, or if the results are what the researchers expected, and the likelihood of publication. So we see if the results are strong, so there's significant results in the direction that we expected, the likelihood of publication is quite high. So it's likely to be published in either a top journal or a non-top journal, and very few of these are not written up. Yeah, so less than 10%. If the results are mixed, again, only around half of these papers will end up being published. Half of them will not be published, and around 10% will not even be written up. And if the results are null, then the likelihood of actually being published diminishes to 20%. And we see that most of the researchers decide not to write up these results. So the file drawer problem is actually quite a large one when we have null results. And again, this probably also depends on the review process, because one of the reasons they're not written up is because they think they have lower chances of publishing those results. Another way to look at this problem is to see kind of what kind of statistical tests get published. So here are some z-statistics from articles that were published in 3 top journals in sociology: American Sociological Review, American Journal of Sociology, and the Sociological Quarterly. And because this is a two-sided test, we know that the value of 1.96 is the cutoff point for finding a significant result. And if the likelihood of having a significant result would not be associated with publication, then we wouldn't expect any relationship. We would just expect, kind of random noise here, and your z-statistics that does not have an impact on likelihood of publication. On the other hand, what we actually see is that if your test is just above the threshold of 1.96, the likelihood of publication increases significantly. So we see that you're more likely to get published if you have a significant result. And to kind of validate this, we can also look at the situation where we have a one-sided test, and then the z-statistic of interest is 1.64. And again, we see this clear cutoff point where studies that have z-statistics that are just barely significant are more likely to be published than those that are not. So again, this is another piece of evidence that we have an issue with publication bias. Okay. So this is one issue, what actually gets published. And again, this on the one hand depends on the researchers because they're less likely to publish what's null findings or findings that they think are not interesting. And then an issue with the review process that some articles are less likely to be published. And then we have another fundamental issue with research, which is specification bias. And this issue is that some research or some results are just more likely to be kind of selected to be in the article and be published. Okay, and this depends on what's also called research degrees of freedom. So the idea is that you as a researcher, you can make lots of decisions, and these decisions will impact the results. And some of these decisions are arbitrary. So you just decide to use a particular method or to filter some cases out, and those decisions can impact the results. And in principle, you want your results not to depend on arbitrary decisions that researchers do. And this, because it's such an important issue, has actually lots of different names, from p-hacking, specification search, phishing, data dredging, and so on. So there's lots of different names for this. And again, this is kind of common knowledge. We saw even in one of the videos before. We know this is an issue that a lot of tests and analysis are done, and then what's being reported is only a small part of that. And again, the selection of what's being reported is probably not random. Again, there's probably particular reasons why something is more likely to be selected for publication than others. And one of the issues or one of the reasons for this is motivated reasoning. So even if we might have some hypotheses or some kind of expectations at the start of research, we might change depending on the results we get, and we might start to believe that we expected this from the beginning. We actually expected to find these strange results, and we argue to ourselves that this is what we thought all along. And again, this is just partially about how human reasoning works and the kind of biases we have as humans. And this issue is problematic when we do lots of tests and analysis, especially when we have small data. So there's a nice example from Christensen and colleagues, where they simulated some data. They had, I think, 3 different groups. It was a relatively small data set. And there was no relationship between being in a particular group and the outcome. But then they decided to include different decisions in their analysis. So for example, if they included dependent variables that were correlated, if they included 2 more observations, if they included the interaction, and if they dropped one of the groups. So you see, as you make some of these arbitrary decisions, the likelihood of finding a significant result increases, even though, again, the truth is there's no relationship here. We expect just 5% of this test to be significant due to chance. And as we combine more of these round kind of arbitrary decisions, the likelihood of finding a significant result increases. And I think this really highlights very nicely how arbitrary decisions lead to false positives in our research. Okay, so I covered the main issues that we currently have. So this issue of selective reporting from the results, and then also selective publication. And we saw that both researchers and kind of publishers and editors are part of the issue or the challenge. So next, I want to talk about some of the solutions that we might have for this. But before that, I was wondering, do you have any questions? We can take maybe a minute or 2 for questions. If you have any, you can put it in the chat. Okay, I'll take that as a no. Okay, so let's move on to the solutions then. Maybe everyone is waiting to hear about the solutions. So I'm going to talk about three main high-level solutions that we have to for this issue. So one, I'll talk a little bit about meta-analysis. Then I'll talk about registration and pre-analysis plans. And then I'll talk about sensitivity analysis. So maybe some of you have heard of meta-analysis, but the idea of this approach is just to systematically summarize all the available scientific knowledge on a particular topic. Again, this is very popular in health research, where there's lots of ongoing research coming up on very particular topics. And then the way this is done is that systematically we search for all the articles on that topic, and then we try to see what is the overall pattern. And the intuition here is that each study might be slightly biased in one direction or another, but if we look at multiple studies, we will see the real pattern, and we see if there's a relationship or not in the data. So let's look at an example. So here, the research question of interest was the relationship between climate change and conflict. And the researchers did a search of the literature, and these are the studies that they found that looked at this relationship between climate change and conflict. And we see that the results vary quite a lot. So there are some studies where they actually found negative relationship, although only one of them were actually none of them were statistically significant, up to positive findings, right? So most of them found a small positive relationship between climate change and conflict. So if we put all this data together, we can actually create the equivalent of a distribution. So the dots here are the main effects from the analysis of these researchers. And then we have the confidence intervals for each of these. And then we can run a model, so something like a multi-level model, and we can get an estimate, looking at all these studies and taking into account their sample size or their certainty or the size of their confidence interval, how certain are we that we have an effect, or what's the overall pattern? And we see here on the right side, this is the distribution of all the coefficients once they're put together. And we see that overall, if we aggregate all of these results, we see there's a positive significant relationship between climate change and conflict. Okay. And like I said, this method is quite popular, especially in health research, but also more and more in the social sciences and psychology, as a good way to summarize current evidence on a particular topic. So on the one hand, this approach is very useful because it can also help us get an estimate of publication bias as well. And the way to do this, one way to do this is to use something called funnel graphs. And this comes from the intuition that if we have a larger sample size, in principle, we expect more precise estimates. If we have smaller sample sizes, we expect less precise estimates. But in principle, if we know kind of the average effect, we expect all of these to be randomly allocated somewhere around the average. And if there's a bias in one direction or another, that will appear in this graph. So let's look at an example. So here we have one of these funnel graphs looking at the relationship between being part of a union and productivity. So this is from economics. And each dot, again, is a coefficient from a study. The dots that are at the bottom, they are studies with smaller sample sizes, while the dots that are on top, these are coefficients from studies that have larger sample sizes and thus larger standard errors. And ideally, what we would expect is this kind of almost normal distribution or kind of funnel where we have approximately the same number of cases on the right and left side of the average effect. And we see the average effect is somewhere around 0 in this case. So this is kind of a good graph. It doesn't seem to show any kind of pattern. Let's look at another one where it's less obvious. So I don't remember here. I think it was the relationship between minimum wage and I think productivity as well, if I remember correctly. But the main thing to concentrate on is the relationship that we see in the graph. And again, each dot is a different study. Here we have a lot more studies. And we see kind of a clear pattern that those studies that have a negative finding are more likely to get published than those that have a positive finding. Even if, again, overall, the effect based on the meta-analysis is somewhere around 0, when we have small studies, those that find negative effects are more likely to be published. So this graph would indicate that in this particular area, we have an issue with publication bias. And this, again, would be an indication that maybe editors and reviewers should kind of consider this when they review and publish articles in their journals. So when to use meta-analysis and when is it useful and what are the limitations. So it is useful when we have a body of literature in a particular area. So there's a lot of research going on. If there's a new topic, maybe there's not enough evidence, so we can't really use meta-analysis. It's useful because it gives us hints if there's publication bias in that area or not. Again, using things like the funnel graph. But this doesn't really solve all the problems that we have. And one of the issues is that it's only as credible as the studies that get published. So if we have issues like peak hacking or this kind of file drawer problem or publication bias, this will also be present in the meta-analysis. So this does not solve that problem, and we need to look at other possible solutions for that. And one of the biggest changes that we can make to improve the quality of meta-analysis is to have pre-registration, which is the kind of next solution I want to discuss. And the idea of registration or pre-analysis plans is to publicly declare what you plan to do before you actually do the research. Ideally, you do this before you actually collect your data, but you can also do it before you analyze the data, so even if the data already exists. And it's really important to have this done publicly and timestamp it. So we have evidence that this really was written up before the data was collected or before the data was accessed. And again, there's a clear trend in this direction, starting from kind of health and then slowly moving in the social sciences as well. So in 1997, the Food and Drug Administration in the US made pre-registration mandatory for clinical trials. In 2000, a website for pre-registration was developed, the clinicaltrials.gov. In 2005, most top medical journals publish only registered trials. So if you want to get published in the best journals in the field, you need to register your trial in advance. And in 2007, also the World Health Organization developed a register for trials, for international trials. Okay, so clearly there's a move in this direction in the health research. And again, similar to what we saw with the time-sharing experiment, having information about registration helps us see what kind of bias we have in publication of results. So here, for example, we have a small number of studies that were approved by FDA for antidepressants. And these are, so the trials were registered and approved, and then we have the results of the trials per se. So we see if the results of the trials are positive, then the likelihood of being published and agreeing with the FDA is very high. If the results are questionable, the likelihood of publication is almost 0. And if they're negative also, there's very small chances of publishing. And again, because we have this pre-registration, so we know all the trials that start collecting data and analyzing it, we can get an estimate of publication bias. And again, we see a very clear pattern that studies that have null findings or findings in the opposite direction are less likely to get published. Okay. And again, we can actually calculate now the amount of publication bias that we get. Looking at the social sciences, also there's been kind of websites or platforms developed to help with registration. So for example, the American Economical Association also developed a website called socialscienceregistry.org, where you can register your study or your trial in advance. There was one developed for international development. And also in the same period, the open science framework was developed, which is has a very big website and platform where people can register their studies and timestamp them and get DOIs and so on. So again, there's multiple platforms being developed to encourage people to do this registration and pre-analysis plans. So this is quite old now, but you see a steady increase in the use of this social science registry website. And you can actually download the data of how many of the studies are on the website and see how this has changed in time. So this is publicly available. So we looked at registering studies in advance. Another thing that you can do is a pre-analysis plan. So this is basically writing up a plan for your analysis before you see the data and you start doing any analysis. And the idea is in this document that again needs to be published and timestamped, you have a detailed description of all the analysis that you're going to do. The kind of things that you want to include are the study design. So for example, how many cases you're going to analyze, what questions or data was collected, what is the study sample, who was included, who was not included, response rates, and so on. How did you measure the outcomes of interest? If you're going to do multiple tests, then you can do families of means effects. So that will take into account the multiple tests that you're doing. Similarly, if you're doing multiple hypothesis testing, you can say in advance how you're going to adjust for that. There are different methodologies to deal with this issue. You can say in advance what subgroups you're going to analyze. Again, this could actually impact considerably your findings and the potential for specification search. Ideally, you should also say what's the direction of the effect. So instead of just stating a general research question, you should say what is the direction in which you expect the relationship, if you have enough knowledge to say that in advance. And you should say very precisely what are the statistical models that you're going to run, what are the variables that you're going to include, what's their sequence and so on. So as much information as possible about the model that you're going to run. And then, like I said, what's really important is to make this publicly available and timestamp it. And like I said, there's actually lots of different platforms for this now. So I mentioned the American Economic Association, Open Science Framework, but also you can do this using GitHub. And if you want, also you can use Zenodo, which is a service that gives you a timestamp and a DOI for any project you have on GitHub. So what are some advantages or strengths of doing pre-analysis plan? Well, the main strength of this is that it limits specification search. So it means that you can't do any research you want. You have to stick as much as possible to what you have written up in advance. It is still possible to do other exploratory analysis, but this needs to be explicitly stated. So now we can clearly separate what is real hypothesis testing and what is exploratory work. So if you want to do new types of analysis, you can present that, but this has to be clearly separated from the really confirmatory work that is included in the pre-analysis plan. Another advantage of doing this is that it actually forces you to think about your expectations. So it forces you to think, what are the hypotheses? If you expect certain relationship, what is the direction of those relationship? And if you already know that, then you can actually have more precise tests. So instead of doing 2-tail tests, for example, you could do a 1-tail test, and then you'd have more power for the sample size you have. Okay, and finally, doing this increases the credibility of your research. So again, this is really important because it proves basically that you're doing confirmatory work and that you had this hypothesis before you have seen the data, and increases your chances of being published afterwards. There are some limitations of this approach as well. So in some fields, maybe we don't have clear hypotheses developed, or some data is very messy and we can't say in advance exactly what we're going to do. So there is some value in doing exploratory work and research, and sometimes we can't write up, a clear pre-analysis plan for exploratory work. Another issue is implementing this when we have secondary data. It's very hard to prove that you didn't look at the data when this might have been available for a few years now, and in principle, anyone could have looked at it. So it's very hard to prove that you are really doing confirmatory work when you're using existing data that anyone can access. And there's still, ongoing debate about best practices when you do exploratory work or secondary data analysis. And I think there are some practices that are coming up as things that we could do even in these situations. So for example, when we do secondary data analysis, we could report if we looked at this data before or not. And again, the people that, organizations that share the data, such as data archives, could also make a report saying if you access this data in advance or not. Other things that are possible is, for example, to split the data and have some data that is saved for confirmatory work and some data that is used for exploratory work. So this is something that is often done in data science, where you have cross-validation. You could also have access to some synthetic data where you could, for example, do some exploratory work and then have access to the real data that you could analyze afterwards. So I think there are possible solutions to this issue of how to do real exploratory work, and also try to limit the specification search, and also how to do this with secondary data. There are solutions for these problems as well. Another interesting concept in this area is this idea of result blind review and reviewing registered reports. So the idea is to move away from traditional publishing, where you write up your article, you submit this to a journal, it gets peer reviewed, and then it gets published, and to divide this in 2 stages. So in the first stage, you might have a results-free review. So this could be something like a pre-analysis plan. This could be peer-reviewed and could be accepted in principle or not by a journal. And then you could collect the data or access the data if it already exists, follow all the stages that you had in your pre-analysis plan, and then submit the final paper for review. And ideally, this should be in principle accepted if you follow all the steps that you mentioned in advance. And again, this would avoid some of these issues with publication bias, because in principle, it doesn't matter if you have a null result, you are still publishing the findings from the study. So this is, again, a solution where actually the pre-analysis plan can be reviewed, and then to help increase the chances of publishing regardless of the findings from the analysis. Finally, the first solution I want to discuss is sensitivity analysis. And this is something that as researchers, we've been doing and we've been trained to do for a long time. And the idea is to do the analysis under slightly different assumptions and see if we find the same results or not. But the issue is that when we do this, it's typically limited in scope. So we might do this because we want to try different assumptions, or the reviewers might tell us to try a different type of model to see if it still works or not. And that might help us to see if we have consistent findings, but it's not really systematic. It's again very arbitrary. What are the things that we test when we do sensitivity analysis? So a solution to this is something called the specification curve. And the idea is to do systematic sensitivity analysis on your data. And this means you have to think very carefully what are the important decisions that you have done, and then to try to run your analysis under these different conditions and see if you find the same results or not. So let's look at an example. So imagine you have 3 important decisions that you had in your analysis. So one, if you dichotomized a continuous variable or not, if you drop the outliers or not, and if you use a cluster standard error or not. So if we look at these 3 decisions, we have 8 different possible combinations of models that we can run to see if we get the same results. As we work with more complicated data, we can imagine that there are actually a lot of different decisions that we can take, and then the number of possible models that we have to run becomes very, very large. And when that is the case, what you can do is actually you can sample some of these decisions and to analyze a subgroup. And again, the idea of using a random sample is to eliminate kind of the arbitrary nature of sensitivity analysis and to randomly search in the kind of your decision making and see if that impacts the results or not. So what are the steps to run a specification curve analysis? First, you need to define what are reasonable specifications. So again, we make lots of decisions, and some of them are debatable, and some of them we think are more clear-cut. So we can think what are the important decisions that we have made, and what could be alternative to those decisions. Once we have all the different specifications we want to run, then we can estimate the model under different specifications, and then we can create a descriptive curve of the results. And I'm going to show you an example of that in a second. And then you can also do a systematic analysis, a little bit like a meta-analysis, where you analyze all the results that you've done in all the different specifications to see if you have a systematic effect or not. I'm not going to talk about this. I'm just going to show you some of this descriptive specification curves that you can make. So what are reasonable specifications? And again, this is not always clear cut. So there is some degree of kind of researcher knowledge and involved in this and decision making. So first of all, you need to decide what are possible or plausible kind of models that are consistent with theory. So for example, would it be possible to have both a linear and nonlinear effect? Would that make sense given the knowledge we have in a particular area? Again, what predictors to include in a model? There could be debates, but again, if there's previous research showing that some predictors need to be included, then we can use that as a way to decide if that's something reasonable or not. Then when we make a list of all possible decisions and combination of models that we can run, we need to think if all of them are statistically valid, because maybe it doesn't make sense to, I don't know, use a nonlinear model for when you do a logistic regression, or depending on the model you're going to run, there might be some combinations that don't make sense either theoretically or statistically, and then you don't need to run those. And then if there are combinations that are redundant, again, you don't need to run them. So, the order of the predictors included in the regression does not have an impact, so you don't need to run that in different ways. So if you have these things in mind, then you can create a pool of possible models that you can run to test if the results you have are consistent over different specifications. So let's look at an example. So this is from a paper that got quite some attention some time ago that was looking at the relationship between the gender name of hurricanes and I think the number of people that died as a result of the hurricane. And here they look at 5 different key decisions in this analysis. So which storms to analyze, how to define femininity, what covariates to include, what type of regression model to run, and how to treat femininity, so the function form of this variable. So there are multiple decisions in each of these, and when you combine all of them, you actually have 1,700 specifications here. This is quite a lot. Again, depending on the type of model you run, if it's not plausible to run all of these, then you can subsample and run only part. But I think in this example, they did run all of them. I'm going to show you this graph in 2 parts. First, I'm just going to show you the top part of the graph. And here, these little dots, blue dots, I'm not sure how well you can see them. These are all the coefficient of interest. So the relationship between the gender and the number of extra deaths due to the hurricane. And we see that this line here is 0. So this means there's no effect. We see most of the analysis show there's a positive relationship between the gender or femininity of the name and the number of deaths, although there are some that are negative. These are only 20 out of 1,700, so they could be due to chance. Then the black dots are those that are actually statistically significant. So we see that out of all of these models, only 37 of them are significant. The vast majority of them are not significant. So they're blue, meaning they're not significant. And the one that was reported in the original paper is actually this large black one. So we see that this is not atypical. So it's somewhere kind of in the middle of the positive range of relationships. So overall, if we look here, it would indicate that there might be a relationship, although most of the times this is not significant. But if we treat this as a meta-analysis, it does seem to some evidence of a positive relationship between gender, the gender of the name, and the number of extra deaths due to the hurricane. And this is how the full graph looks like. So here on the left side, we have different decisions that are made. For example, here, what are different variables they are controlling for and in different interactions. So for example, this is interaction between time and damage and year-end damage or no interaction at all. Here we have how the functional form of damage, the model that was used, how the femininity was coded, and then other decisions. And again, each dot represents one of these coefficients. So we see that, for example, the negative coefficients that we have here appear when you have a particular combination of, for example, the model that you're running and the way you define damage and the way you define femininity, all of these together combine to have this atypical result with a negative relationship between gender and number of deaths due to the hurricane. So the takeaway here is really this idea of thinking systematically, what are the key decisions that you made, trying to write those up and think what are all possible combinations of those decisions that you could run in your model in order to do a sensitivity analysis. And then finally, running all of those and doing a graph like this to see what are the patterns that you see and to understand if there's a systematic relationship or if what you find is just noise due to particular decisions you have made. Okay, so maybe let's take another short break here and let me use this opportunity to ask if there's any questions. Okay, so there's a question about synthetic data, which I think is really good. So how is synthetic data useful for doing reproducible work? So one example of this is that sometimes a challenge with doing pre-analysis plan is that there are issues with the data that you don't know in advance that you need to deal with. So there might be outliers or a strange distribution or multicollinearity or lots of arbitrary things with the data that you can't guess in advance. And when you start looking at the data, you need to do something about them and they might end up being arbitrary. So one solution to this is to give synthetic data where researchers can look at it and see what are the distributions, how much missing data there is, and try to think what are the important decisions they need to do in data cleaning. And this is the important part, without looking at the relationship with the outcome. So for example, you could give synthetic data without the outcome, or you could include some noise in the outcome. So the decisions regarding data cleaning are not influenced by what you think is the overall result. So you're not kind of cleaning the data, so you get a positive result. So that's one way in which I read about this idea of using synthetic data. The other advantage of using synthetic data, and I'm going to talk a little bit about sharing data, because this is really important for replicating work. And some people may not be willing to share their data because they think it's sensitive. And having synthetic data is a compromise where you can share the data so other people can replicate your work without, the issue of identifying individuals. So that's a way in which you can use synthetic data to help with reproducibility. Jamie, so I hope that kind of answers your question. Then we have a question from Daria. So this question is about the funnel graph. So the question is, how do we know that lower frequency of published studies with positive result is not due to the scarcity of studies that find a positive association, but because of publication bias? So the idea with the funnel graph would be, let me see if I can go back. We assume that this average, this line here, this average, or this, you can think of it also like a result from a meta-analysis. This is the truth. And again, might not be correct, but we assume that if we have all the studies and we put all of them, this is as close as we get to the truth. And then when we have smaller sample size, the idea is that arbitrary decisions can have a bigger impact in this situation. So, you have smaller sample size, the likelihood of doing multiple tests and finding something significant increases. And then again, this could be related with the publication bias. So the idea would be that if the assumption that this average is the real score, we would expect to have an equal distribution on even when we have small samples. But because we have small samples, and then these arbitrary decisions have a bigger impact in that case, and we see this bias towards publishing either positive or negative findings when we have smaller samples, it would indicate we have a publication bias. Again, it's not clear cut because we do make these assumptions that, well, this is the truth, and that might not be the truth. Maybe this distribution is way biased because of not including any of the null findings, for example. But we would expect that studies that have small sample sizes, in principle, they should be randomly allocated around the average. So there's this strange pattern that is happening. And again, it might be due to this higher chance of publishing and higher chance of finding basically false positives or false negatives. I hope that explains a little bit. Again, it's not perfect because it makes some strong assumptions about how this would work, but statistically speaking, you'd expect all of them, they should be randomly allocated around the average. And if they're not, then there's a bias, and that's more likely to happen when you have small sample sizes. Okay. Okay, and then Paul had a question. So Paul is asking about journals giving credit when things are done the hard way. Yeah, so I'm going to talk a little bit about journals in the next part. So I think there's work being done in journals, and it comes through the standards that you use and through the fact that they might, on the one hand, raise expectations, so it makes it hard for everyone. So you need to share your code and your data, and on the other hand, has special space for example, publishing replication studies. So I think there's work being done in both of those directions, and some of it I'll cover in the next section with this idea of the standards that are being introduced in publishing. So I think that's a really good segue to the next part.